These cats will be sort of different shapes and sizes based on what we're doing and the difficulty of the subject and various other types of factors. But nevertheless, what we want to go ahead and do now is just sort of talk about the answers, have a discussion about the answers and the questions, particularly those that might have been more difficult. As this relates to the examinations, of which the first exam is next week, these types of questions that you saw here and then those that I passed out last week will sort of be the basic format of those questions that will constitute a minority of the exam. They're not going to be incredibly widespread, but will be a section of the exam that will allow you to demonstrate your ability to apply some of the material in ways that are a little bit more difficult than simply recalling terms or demonstrating that you understand the content. So again, this is sort of how we scaffold, or this is how we delineate those that have an exceptional, outstanding grasp of material versus those that have an average or above average comprehension but haven't developed that acumen in order to really apply the knowledge in an incredibly critical and meaningful way. So with that in mind, let's start talking about these. All right, so the first one, ionic versus covalent bonds. I sort of touched on this briefly earlier today, so hopefully that was helpful. So, in terms of that difference, right? So, how do you describe how ionic and covalent bonds are different? Well, again, ionic, you can talk about difference in terms of strength, right? So, what's the stronger type of bond? Covalent, right? And by stronger, that means that they're actually, the reason why it's stronger is they're actually sharing bonds. They basically have a physical interaction with each other. They're basically shaking their hands. All right? So, it's going to take a lot of energy to break that chain. Whereas an ionic bond, you basically have two atoms, one of which is donating an electron to the other. And when you donate an electron to your little friend, so in terms of salt, for instance, right, which is sodium chloride, sodium likes to, because of its valency, because of the fact that if it gets rid of an electron, then its outermost shell is filled and it's happy. It's more than willing to give an electron over to chlorine, which is almost at the opposite end of the periodic table, that just needs one more electron and its outermost orbital is going to be filled, so it's going to be happy. So they have this relationship where sodium just basically punts an electron, throws an electron over to chlorine. And as a result of that, because then you have this inequity between protons and electrons, sodium now has more protons, so it's positively charged. Chlorine has more electrons, so it's negatively charged. And just by that phenomenon, because those are opposite charges, they're going to attract to each other. And that's what really makes salt a compound, a molecule. Right? But there is no handshake here. Those electrons, this electron is basically being completely given to chlorine. Whereas when you have two carbons with each other, those electrons are going back and forth, back and forth, back and forth, back and forth at 1,000,000 miles an hour. And as a result of that, that gives the characteristic of a covalent bond being much, much stronger. And in terms of what the electrons, how the electrons are behaving, the electrons are being shared across both atoms in the covalent type, whereas they're being donated or received in the ionic type. Okay? So that strength can be applied in a variety of ways. You know, is it more difficult to break salt or break sugar? So sugar is more difficult to break down because sugar is made up of a bunch of covalent bonds between carbon, hydrogen, nitrogen, in some cases, oxygen. So you would need exert a lot of heat to break down sugar as opposed to salt. which is readily soluble in water. Right? Even if you take a cold glass of water or water at room temperature, right, you add a certain amount of salt, the salt will instantly dissolve because it's a weak bond. That ionic bond is not incredibly strong. But in terms of sugar, unless that coffee is hot, you take cold coffee, hot coffee, if it's cold, that sugar is just going to settle down to the bottom because there's not enough heat in it to break down all those covalent bonds that are keeping the sugar intact. All right. Now, all right, hydrogen bonds, van der Waals interaction. So this would have required you to remember that jargon, remember that terminology. So, hydrogen bonds, van der Waals interactions are both what are known as sort of these weak interactions amongst atoms. And they don't even constitute a sharing of electrons. So in terms of scale, covalent bond is really strong, ionic is weaker, but hydrogen bonds and van der Waals interactions are much, much weaker than even ionic bonds. These are bonds that are breaking and forming 1,000,000 times a nanosecond in a glass of water. And why are they so weak? Well, because they're not even involved, an electron isn't even being donated or received or shared in these situations. So again, in terms of hydrogen bonds, what are we talking about there? What needs to be present in order for hydrogen bonds to actually even be involved, for hydrogen bonds to even be able to form? What other types of bonds need to be around? Any ideas? Let's say they're covalent. What do you think is going to form hydrogen bonds? Non-polar covalent bonds or polar covalent bonds? It's going to be polar, right? And why is that? Well, again, let's take our friend, let me grab another, see if one of these is better. Okay, let's take that dynamic. Again, we talked about this being a polar bond, oxygen and hydrogen, right? Oxygen's the bully, it's the ball hog. So you have this partial, this delta means a partial charge that's negative. Hydrogen has a partial charge that's positive due to how much the electron resides on one side versus the other. All right, so if you have another OH group around, you have another OH group in close proximity. The same dynamic exists between these guys, right? Partial negative, partial positive. If they're close in proximity by chance, then this partial negative is going to be attracted to the partial positive in both these directions. And that creates what's known as a hydrogen bond. All right, so there's an attraction there. It's not incredibly strong by itself, but if you're thinking about in a glass of water or a DNA molecule, the fact that there are millions of these bonds formed, collectively, additively, they represent a force that has a significant impact in how life works. So again, the classic example of this is double-stranded DNA that we'll look at in much more detail later on. But the reason why those strands come together is because those nitrogenous bases have or form, they stick out of the DNA molecule. So each one of these little bars here represents a nitrogenous base that's sticking out of the backbone of the deoxyribonucleic acid. And there are nitrogenous bases there that are constituted with oxygen to hydrogen bonds, with polar covalent bonds. So what attracts one DNA strand to the other DNA strand, what makes this, if that's red, attracted to this yellow and make this chain, make this linkage, is the hydrogen bonds that form between the nitrogenous base on this strand and the nitrogenous base on the other. Now, DNA strands in and of themselves are millions of nucleotides long. So as a result, again, the collective force of a million hydrogen bonds makes those two DNA strands stick to each other really tight. Van der Waals reactions are even worse than hydrogen bonds in terms of their strength. They're the weakest of the weak. All right? And they just represent a force that can happen, again, instantaneously, 1,000,000 times a second within atoms, where you, by chance, even in a nonpolar covalent bond, which this would represent, carbon to carbon, same atom, or same element, again, within a nanosecond, a quadrillionth of a second, there might be a circumstance in which it just so happens one more electron is on this side of the bond as opposed to this side. So you're talking about the tiniest little positive charge and the tiniest little negative charge. Now again, in that one instance, that's going to mean nothing. It's going to have no impact. But in the case when you're talking about very complex molecules, these are the types of interactions that can have direct impacts on, for instance, the ways in which proteins are formed, which you'll get into in these next chapter questions. So again, the difference between potentially a protein looking like this or looking like this, just a big glob or a string, can be impacted by Van der Waals interactions and hydrogen bonds, just because there could be so many of them together, they could have an impact. All right, buffers. All right, so again, this relates to pH and the propensity for most of life to try to stay as neutral as possible, or somewhere close to neutral. Most life exists basically on a scale between 6 and 8 in terms of pH. There's some exceptions. There's bacteria called acidophiles, and there are alkalitophiles that like really acidic conditions or really basic conditions. But by and large, we want to stay, we being life, want to stay relatively neutral. So how do we do that? Well, we utilize compounds that are known as buffers. Right? And this figure from the book talks about one of the buffers that we find in our own bodies, that we find in our own blood. It's called bicarbonate. So this little guy is bicarbonate, and what basically it does is that and remember, what's pH about? It's about the number of hydrogen ions, or the number, and that compared to the number of hydroxyl ions. So, in a situation in which, for whatever reason, your blood pH begins to spike, and by spike I mean it starts to go down because there's way too many hydrogen ions, all right? This bicarbonate can suck those hydrogens up and form compounds that basically that shield those hydrogen ions and hydroxyl ions away, so that the pH basically then becomes normal again. So, it can raise the pH. So the blue arrows here represent the process of raising the pH as more carbon dioxide is made. So an outcome, a product of this carbonate trying to maintain a relatively equal number of hydrogen and hydroxyls, is often the production of carbon dioxide, which we ultimately do exhale through our lungs, right? So this type of mechanism is incredibly important for life, as I said, and to answer the question, helps to prevent the drastic swings in pH. So it's going to suck up hydrogen ions if they're too high. It's going to suck up hydroxyls if they're too high, and that's ultimately going to create an environment in which the number of hydroxyls and the number of hydrogens are relatively equal, which is how life wants to live. All right, what makes carbon so great for organic life? Okay, so somebody mentioned it being nonpolar, or some group. So in terms of it being nonpolar, yes. Oh, I did skip one. Okay, yeah, thank you. So number 4 is about insects on water. All right, so what property of water? So this is trying to figure out whether you understand the properties of water that make it such an amazing molecule. And water is really weird as a molecule. It has very strange behaviors compared to others. All right. In terms of this question, one of its strange behaviors is the fact that it is so polar that it likes to adhere to itself. So water molecules like to stick together because of those charges we talked about, because of those hydrogen bonds that form. All right? And the way that they do that in liquid water, particularly where you have the interface of liquid and gas, right? So at the top of the ocean or the top of a lake or a river or a stream, you have this phenomenon where you have gas and you have liquid, this interface. And particularly right there at that line, all of these water molecules are stuck together by hydrogen bonds. They create basically this surface. In so much that, okay, and that creates something called surface tension. So it's like this really, I mean, obviously it's not incredibly strong because we can't walk on water. At least I can't. But it's enough for insects and very small organisms that have very low density to be able to survive, or to be able to actually maintain themselves on top of that water. All right? I mean, what's another example of the fact that we know water is, the adherence of water creates a lot of surface tension? I'm sorry? So that's tied to their polarity and the fact that they are that the wax, typically water droplets form when it's on a surface that is hydrophobic, like a car, right? Car, water beads up, that's because there's wax on the car, or the wax itself is made out of lipids, okay? Well, just think about, somebody jumps too high off of something, lands into the water, does a belly smack, or worse, dies as a result of that. It's water, it's liquid, right? It's soft. So what forces basically the organs of the body to be crushed and massive trauma to occur if you jump too high off of something and land into the water? Okay? This is like hitting, a line of bricks if you get too high. Part of that is the surface tension that's created at the layer of the water, because those waters adhered to each other. So if you hit that at such a high rate of speed, it can cause a traumatic event in the body. All right, now 5. Oh, yes, of course.
Is that kind of what allows people to dive off a ladder into like a very thin pool of water? That surface tension and the water buildup allows that person to just like hit the water but not necessarily kill themselves because of the lack of water?
Right, so you're talking about diving, the way
like
you hit a water.
People, how they
Sure.
go to the top of a ladder and then jump into like a pool with like this much water from the top of a ladder, and how they just don't kill themselves from that.
Right, so part of that, again, so part of that as well is how do you spread the pressure? So the insect, in that picture, the insect's legs are very far away from each other, right? So it's, even though it weighs a certain amount, even though it's so dense, it's positioned, it's anatomically evolved in a way to allow the pressure to spread. So based upon how one hits the water or dives into the water, if you limit the amount of surface area that actually touches the water, then that correlates to just how much of an impact will occur when you actually hit the water itself. Right? So basically when you see a dive that's rated in a 10, that's because that diver has hit the water in such a way that's minimized the amount of skin that hits that water initially as much as you can, as opposed to just jumping in and landing on your stomach, which basically is a significant amount of surface area hitting that top of the water all at the same time. All right, 5, carbon about organic life. So what is it about carbon that makes it really useful? Well, again, it forms nonpolar, it can form both polar and nonpolar bonds, right? Obviously, if it binds with itself, it's going to be nonpolar, but that phenomenon of the fact that carbon and hydrogen, when it bonds to hydrogen, that remains nonpolar, is incredibly important for life, because those carbon to hydrogen bonds make up a really important part of our cells that we'll see later on in a couple weeks. It also can bind to how many bonds can carbon make with other atoms? 4. So that process of actually being able to make 4 bonds, okay, allow carbon to be configured in molecules and make different types of shapes in very significant ways. It can bond with itself, right? It can create double or triple bonds in some cases, which also delineate certain types of molecules, like saturated and unsaturated fats. So all of those pieces of carbon, all of those characteristics of carbon play out to make it really important as it relates to how life works. Okay, so number 6. All right, so this one was pretty tough, right? But again, I wanted also to get you to continue to think, like in chapter 1, about the scientific method and how you would utilize some of those particular applications in a way that is helpful. All right, so we're talking about unsaturated, saturated fatty acids, right? So what's the delineator there? I just told you it's about, and what delineates those two is going to be whether or not any of the carbons have formed double bonds with each other, right? So in one circumstance, the fatty acid all you're going to see are carbons singly bonded to each other. In another circumstance, you're going to see maybe just one instance in which 2 carbons have double bonds with each other. That's the determinant between an unsaturated and saturated fat. All right? So, which one's which? Okay, so saturated, no double bonds. Unsaturated, at least one double bond. Okay, so why is that important? Well, because of that phenomenon, again, the way molecules are bonded to each other has an impact on how they behave. So it also impacts the ways in which that they their physical state of matter at room temperature. All right, so we go back to the question at hand. Seeds of temperate plants tend to contain unsaturated fatty acids, while the seeds of tropical plants usually have saturated acids. Okay, so tend to have saturated fatty acids. Okay, so what we know is that saturated fatty acids, what you would need to remember, what you would need to recall, is that saturated is solid at room temperature. Unsaturated is typically liquid. Okay, so these seeds, depending on what type of fatty acids they have, that can dictate whether they're harder or they're sort of more liquidy in a sense, their relative densities and the like. Okay, so again, how would you test your hypothesis? Well, and again, temperate plants, we're talking about plants that are from climates that are cooler, right? That have seasons, that are farther away from the equator. Whereas we're talking about tropical, closer to the equator, okay, temperature deviation doesn't occur very much. It's relatively warm, it's relatively wet. Okay, so the question is about, okay, so how would you test your hypothesis? All right, well, you could potentially do it in a number of ways, right? So basically you would need to form some test in which you're taking these seeds from these different locations, right? And somehow you're assessing which ones are saturated and which ones were unsaturated. So if you know the physical state of room temperature, your test could be involved with actually extracting those fatty acids and seeing if they're solid or they're liquid. Or if you got really technical, you could talk about looking at the atomic level and looking for the presence or absence of double carbon bonds, double covalent bonds, which would be, again, the determinant in making those happen. Okay? Okay, so if that hypothesis is supported, all right, that we see saturated or solid and that unsaturated are liquid, okay, what does that mean? All right? So did anybody come up with a hypothesis or a conclusion based upon that? What would we go back to in the question to try and it must be a link, right? It must have something to do with climate. Right? So in a certain sense, if saturated, we found saturated are going to be more in tropical, unsaturated are going to be more in temperate. Okay? Any ideas? Anybody come up with an answer that they're willing to share? All right, so in terms of this as well, typically it ties into how the seed is going to try to survive in its initial attempt to sprout, to become an actual plant. So in tropical conditions, it's helpful to be more solid as a seed, whereas in in temperate conditions, having that liquid is often more important. A lot of this ties into, again, in cold weather, having a more liquid surface or having more liquid within the seed itself, okay, can protect from, can insulate the seed and protect it, whereas in terms of the tropical, it's usually harder, which protects the seed itself from some of the humid conditions and warmer climate that exists closer to the equator. So again, anything that would tie to, well, and again, I wouldn't necessarily expect you to know that. Again, I just sort of threw that in there to try to get you to think about these things. But anything tied to, again, if you have different things producing different substances and they're from different environments, then there must be something about those environments that's driving the ways in which these organisms have evolved. Yes?
Why does the liquid fat keep it, what's the word you use? Insulated better than a solid fat?
Right. So, so solids usually conduct heat, right, at a much higher rate. So the liquid itself, when I say insulate, what I probably, the better word I should have used is that the liquid itself allows for the seed to, trying to think of a better word than insulate. So when you have a seed that is high liquid content, high fatty liquid content,
it maintains its temperature more.
Right. So
because liquid has a higher heat, specific heat capacity.
So think about oils. All right. So think about olive oil or peanut oil or those types of oils, right? What makes them so great to cook with? Because you can generate a lot of heat and it stays liquid basically, right? It doesn't evaporate, it doesn't smoke up, right? So in that case, think about it in that capacity. It can absorb a lot of heat and yet still not evaporate, and those types of characteristics will be beneficial.
Okay.
All right, good. So we got about 30 minutes left. We can take as you go back into your teams, we can take a quick break, but before we do that, you can take a 5-minute break if you need to use a restroom or walk or things of that nature. But for our chapter 3 piece, we're going to do something also that we will do from time to time. I'm going to divvy up the questions so that you're only going to be responsible for, your team is only going to be responsible for probably 2 or 3 questions, and we'll focus on those as we go to the next stage of this. That'll allow us to save a little bit of time. We'll reduce the amount of time you'll have to about 15 minutes or so, but you're only going to be responsible for about 2 or 3 questions. Okay? So you can go ahead and go back into your positions. If that's across the street or across the hallway, go ahead and do that. And I will be around to give you your assignments for what questions you're going to be responsible for answering.
Minutes today.
Right. So I'll get your name on this, so that'll take care of it. I'll see your names on this. So
Okay.
we'll get to go. Yeah, that'll be the way to do it. All right, you all have seen one. Okay, so I want you to focus on question 2, question 4, and question 7. You all focus on question 3, question 5, and question 8. You all focus on questions 4 and let's see, questions, these are good, right? Questions 1 and 4 and 9, I'm sorry. 1, 4, and 9. 9.
There was one where I was like not sure for a little bit, but then you said each one.
Sorry about that. You have more time, I promise.
appendix and different animals.
Yeah, there's like because like he died.
sorry. Okay, dehydration synthesis is when like there's a compound and then I don't know what happens, but it breaks down into like one molecule, an element and then water.
So that would be the nitrogen part then. You said it was breakdown.
I don't know what transplant
I was talking about just like my tongue did that thing where like it stops doing good.
Oh, okay.
Alright. We got
it's about the bonds like breaking down and like or like coming back together, right? So hydrolysis is the opposite. It's like when it's like water and an element come together.
You start with the sodium and protein and work back into a smaller parts of your amino acids. I could be wrong. You hypothesize that the enzyme will no longer be able to catalyze a specific chemical reaction it was made to.
no.
on how to do that. Well, yes, I Googled it. It helps.
It's sort of, basically, they have the same.
I didn't know
mindset
might,
that
it
we're
might not
doing
turn out how I
everything.
want it.
The
I didn't know there was a
starts with
method
the
to it.
general idea,
I just
like
thought
the
you
main
threw it
thing
out.
that you know already, and it figures
Because
out
usually
what
I'd be
you
like, I don't know,
and
usually they
everyone
look
starts
like
with
a
little
knot.
things, and you kind of work up to a general.
Okay,
what those
sorry,
things mean.
my natural hair is kind of a mess.
Like works backwards. So the way that it was explained to me was
Just take it,
deductive
take your two
reasoning
hands,
is all cars have engines. I have a
like
car,
comb it back
so
into one
my
hand,
car has an engine.
pull it over.
And inductive reasoning is my
Yep.
car has an engine,
There you
this
go,
car has
that's
an
it,
engine,
and
her
then you
car
make
has
it
an
look so
engine.
easy.
So all cars have
I don't
engines.
like how it looks. It probably looks retarded right
So
now.
the
You can't see it, but
deductive and inductive, the way that
no, I don't care.
was deductive start, you start out knowing all cars have engines.
Yeah.
So I have a
Cause one of my
so my
coworkers
car
has it, and I'm like,
but
I don't have it. My hair was
I don't
longer
have
at
the
work,
character
well, but like now,
her car
yeah,
has
just.
an engine. This car has an engine, so all cars must have engines.
Put something.
Sorry.
I can't say that it's so cool.
So yes, you put deductive and basics, because I don't think that they were necessarily trying to make something help people when they figured it out, they were just
Sorry,
trying
I
to
just like
figure
called
it
you
out.
out. That was rude.
Yeah, just to know it, so basic.
Okay. And the bond
I'm
is right
assuming
for
they started out with proteins, working a certain way, so they were like, so protein has to be like this, so it must be deductive, but.
covalent or ionic bonds break when they're sharing electrons.
Okay, so we are, let's see.
Oh, Rachel, sorry.
Biology's just kind of my thing.
What?
We're like,
Okay, yeah. They're on more synthesized through monomers, which can breaking down into.
he said, I asked him to explain it, he said it is certain elements are more electromagnetic than others. So
We
we
never
know
moved out of the way.
if an element is electromagnetic and it's with a different element.
About a minute left.
we know if it's polar or not, right? It doesn't mean that yet. Actually, I know the answer to number 6.
What are you trying to do?
Cause I have them all.
Yeah, it's not easy.
Yeah, I think there, if you go to the middle, he's got these little things called Quizlets. And you click on those and you've got the flashcards.
I wasn't here for
like the little mini tests and
Just
just
like
like
Like it was a lesson.
I like the.
Are you in our chemistry?
Yeah.
You're supposed to do like, you're supposed to wear the same thing.
I brought scrubs. Yeah, me too.
It's called
Scrub
flashcards,
top.
but it's
Yeah,
like
from biology lab's gonna be able on
that
chiller.
is flashcards, but like you
You're
just
not
like
so
flip it and you go to the next one. The other one that'll ask you the question.
That's what
answer it.
I know, cause I swear, I don't even know what's going on. Like, I'm trying to figure out how to work now. What's that to do with dehydration symptoms? I've never done
We would get like detention if we had our phone on us, even if it was off. Something like that. You have to go to hit whatever one we're on, so it's just going to flip the middle and go down. And, it's the ones that
Turn my back.
I don't think it's the same person first. Like I got it on the same.
So that's 2 cases, or is it just the right?
No, it said someone else put a lot of work into it.
Nice catch.
Well, y'all back up some. Right up on them.
All right. Like I said before, I will send these back to you once they're graded to every member of the group. So if you want to utilize these again, you want to see how, obviously you'll see how well you did, but then also if it helps at all in terms of preparation. So let me go through some of these. Well, I'll go through all of these and then we'll talk about the exam a little bit. I didn't give anybody one, I'll talk about one. So one was about trying to create a hypothesis if you have a change in the enzyme. Okay, so again, enzymes are proteins, and proteins are built upon subunits called amino acids. And the amino acids can have different types of characteristics. One of such characteristics is either being polar or non-polar. We already talked about the fact that polarity is tied to what types of elements are bonding to each other. So in terms of answering this question, how could you test for that? Well, again, polarity is going to have a characteristic of being more attracted to water, of liking water, or sticking to water, as opposed to a non-polar amino acid, which is going to be afraid of water, which is going to be repulsed by water. So the scientific terms for that are going to be hydrophilic, which means you're attracted to water, you're polar, versus hydrophobic, which means that you're repulsed by water. So setting up some type of experiment that would test the relative polarity of those proteins, either the abnormal version or the normal version, could be a way in which you could test and discriminate amongst those two types of enzymes. Dehydration, synthesis, and hydrolysis. These are reactions, a group of reactions that basically dictate the formation or breakdown of these macromolecules. And as the names imply, they all involve water. Dehydration is a process that actually synthesizes molecules, that builds them up. So in the process, the dehydration indicates as you're removing water, when you're bringing two subunits together, you smash them together, water comes out, you've dehydrated the compounds, but through that process, you get them to stick together. Whereas hydrolysis is making stuff wet. And if you make stuff wet with energy, that's going to be the opposite reaction, where you're going to break those subunits apart. Similarities and differences between glycogen and starch. What types of molecules are those? Which one of the four macromolecules are those? Carbohydrates, right? And then specifically, to be more specific, they're polysaccharides. So they're long chains of these monosaccharide units. We talked about the fact that glycogen and starch are both used to store energy. The difference becomes exactly how they're connected together. So even though they're rings of glucose, the ways in which the glucose wraps itself around itself looks different, so it takes on different shapes. Glycogen looks different than starch. And then as well, glycogen is a form of energy storage primarily found in animals, whereas starch is the preferred method for plants. Why can't humans digest food that contains cellulose? All right? So again, we don't possess the enzymes necessary to break those glycosidic bonds, those beta 1,4 glycosidic bonds that we saw that break open or that connect the subunits found in cellulose. Now animals, so herbivores like cattle, for instance, they don't possess those enzymes either. So how do they break down cellulose? Well, in their intestinal tracts, they have evolved to create an environment so that bacteria and these other weird organisms called archaea can live in those environments, and those organisms actually are the ones that break down the cellulose for the benefit of the animal. So that's an example of something you'll talk about more in the second semester of this class, which is symbiotic relationships. I scratch your back, you scratch mine, happening all the time in our environment. One example is bacteria that live in the gut of cows and other types of herbivores that allow for the cellulose to be broken down, and therefore the animals can actually utilize that for energy. The bacteria are cool with it because the intestinal tract itself is a really, it's like Cabo for bacteria. It's a really nice, warm, comforting place for them where they can avoid a lot of negativity and potentially dangerous situations. So in terms of lipids, what are some of their functions? Well, we can use it to store energy, okay? We can use it for insulation. It helps keep our bodies warm in some instances as well. And it acts as components of the cell. So in the chapter they talk about the fact that the plasma membrane, that boundary that makes up sort of where the cell begins and ends, is made up of lipids. And that's going to be a really important part of chapter 4, which we'll cover in 2 weeks. 4 protein structures. So again, this is, if you review the chapter, when they talk about proteins, again, we're talking about these chains of amino acids. And there's 4 different versions of that, right? So there's real quickly, there's primary, secondary, tertiary, and quaternary. All right? So, in terms of describing those, it's on a scale of complexity. When you're talking about a primary sequence, you're just talking about the sequence of amino acids that exist on the strand. Think about it as a straight line. It has amino acid 1, 2, 3, 4, 5. And there's over 20 amino acid combinations that there can be for every spot. When you're talking about secondary structure, that involves the ways in which, once you've identified those amino acids, how they start coming together in different ways, okay, so that they can create these sort of helices type of structures, or they can create these what are called beta sheets type of structures, which are referred to in the chapter. So you're starting to make an actual shape based upon the hydrogen bonds and the covalent bonds that are formed based upon these different amino acid combinations. Tertiary is basically taking the totality of these different helices and sheets and making some type of three-dimensional structure. And as we'll see later on, it's that structure that really is going to be the determinant in how this protein is going to function. Is it going to break down sugars? Is it going to provide structure to the cell? Is it going to be involved in synthesizing DNA? All of that is dictated ultimately by the shape of the protein, as we'll see in much more detail later. And quaternary is a situation where some proteins can't work, can't function on their own. They are part of a puzzle piece of a greater puzzle where you have multiple proteins getting together, making interactions, and from that, this multi-protein conglomerate, that is the form that's going to be functional. So the greatest example of that, the easy example, is hemoglobin, which is what carries our oxygen in our blood, right? It's actually 4 different proteins that configure themselves in a way to trap oxygen and be able to carry it to the various parts of our bodies. All right, 7, we talked about the structural differences between RNA and DNA before, so if you had that assignment, you were lucky. I'm not going to go over that again. The 4 types of RNA and how they function, right? So, in terms of that, so we have and again, RNA is sort of dictated is denoted, the different types of RNA is denoted by a letter that sort of serves as a prefix. Okay, so the 4th one's escaping me at the moment. Let's see, which one am I missing? Oh, right, micro. So, we're running out of time here, and I will post basically my, I'll talk about this more on the forums to make sure everybody's clear with it. But these form types of RNA are clearly dictated in the chapter, and again, you should know the function of those and their basic properties. So I will also cover the remaining, let's see, we only have one other one, and I will make sure to cover that in the forum, in the Q&A, so you can see the answer to that. That one's pretty self-explanatory. What you'll also see, since we run out of time, I do want to be sure to allow you to give you some advice in terms of the final. I will do that via video that will be uploaded later on in the course, later on tonight actually in Moodle. That'll give you some perspective as to what I think you should do in order to be best prepared for the exam. Exam will be in Board 100. It will be Scantron based, so make sure you have your Scantrons prepared. We will only do the exam, so you have 90 minutes to complete it. Once the exam is over, you're free to leave next week. And I think that's pretty much it. So again, I have office hours till 4 o'clock. If you need to talk to me about anything else, feel free to do so. Have a great Tuesday. Library should only have one color now. It's in my syllabus. It's in the syllabus. I think it's the red ones now.
No.